One protein helps 75% of cancers to spread, and we may have a way to stop it. The protein MYC is part of healthy cell activity, but when cancer cells develop, it goes haywire, going outside of its normal, careful, controlled role and helping cancer to spread. Now scientists may have found a way to stop this from happening. Part of the problem in reining in MYC is that it's a shapeless protein, one that does not really have a structure that can be targeted. That makes it difficult for drugs to effectively identify MYC and keep it behaving normally. However, a team from the University of California, Riverside, UCR, has been able to develop a peptide compound that can bind or interact with MYC and help get it back under control. MYC is less like food for cancer cells and more like a steroid that promotes cancer's rapid growth, said biochemist Min Xiu from UCR. That's why MYC is a culprit in 75% of all human cancer cells, he says. Normally, MYC's activity is strictly controlled. In cancer cells, it becomes hyperactive and is not regulated properly. The researchers were able to study the small amounts of structure that MYC does have in order to build up a library of peptides that might be able to snag on to that structure. One peptide in particular, NTB2R, proved particularly adept to disabling MYC. The test using a culture made from human brain cancer cells, NTB2R, was shown to successfully bind to MYC, changing the way cells regulated many of its genes and ultimately decrease the metabolism and proliferation of cancer cells. It's a bit like taking someone's hand behind their back, stopping them from doing anything much at all. Key to the breakthrough was the earlier work by some of the same researchers, which recognized that as the structure and shape of peptides were changed, these molecules became better at interacting with shapeless proteins like MYC. Peptides can assume a variety of forms, shapes, and positions. Once you bend and connect them to form rings, they cannot adopt other possible forms, so they have a low level of randomness. This helps with the binding, says Xiu. We improved the binding performance of this peptide over previous versions by 2 orders of magnitude. This makes it closer to our drug development goals, he said. There's still a lot of work to be done, though these early results are promising. Right now, the peptide is being delivered via fatty spheres called lipid nanoparticles, which are not really suitable for dispensing drugs, so that will need to be changed, he said. Rigorous tests in human subjects will also need to be carried out, but we just might have found a method for stopping one of the ways in which cancer hijacks healthy biological processes in order to survive. MYC represents chaos, basically, because it lacks structure, says Chu, and that then is a direct impact on so many other cancers, make it one of the holy grails of cancer drug development. We're very excited that it is now within our grasp, he said. This was published on the Journal of American Chemical Society, and it's on Science Alert by David Neild. Please leave your comments. Thank you for your support. Kindly support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.